Yes. Evolution, cell biology, biochemistry, and developmental biology have made extraordinary progress in last 100 years. Most striking of all of the sudden eruption of molecular biology starting since 1950. Today's esteemed guest speaker is one of such personality who have been pioneer and the inspirational epitome of this molecular biology and cell biology. I would like to take this opportunity to introduce our guest speaker of the day, Professor Dwapan Goswami sir. Professor Dwapan Goswami sir has done his BSc in biochemistry with a certificate in vocational biotechnology from St. Xavier's College in Ahmedabad and MSc in biotechnology from Gujarat University Microbiology Department. His doctorate in biotechnology was earned from Department of Biotechnology at PDPIS Chartered University. He worked as a UGC CSIR Senior Research Fellow in Biotechnology Department at PDPIS Chartered University. He worked as a DST Junior Research Fellow on Project Eco-Friendly Bricks, a new material in civil engineering. He passed in the life sciences. He passed the CSIR net again in December and earned the 36th rank. He has written many book chapters in addition to numerous research papers in high impact journals. Additionally, skilled in animation development. He produced animated YouTube videos for education purposes. For participating in the UGC 2009 research program, Professor Goswami sir received the coveted Joseph Grasses Research Fellowship Award and a gold medal. He has an extensive hands-on experiments on plant tissue culture and molecular biology. I, Dr. Dwani Upadhyay, Assistant Professor, Microbiology Department, would like to invite Professor Dwapan Goswami sir for the today's talk. Please, sir. Respected sir, I request you to kindly start the today's talk, sir, please.
Okay, so yeah, good afternoon, ma'am. That was pretty elaborated description about myself, and I don't have words, but probably it was too lengthy and too elaborate. Yeah, but anyways, thank you. And I take this opportunity to be a part of this webinar series that has been organized by Parallel University. And I'm pretty much thankful to the organizing faculty members for calling me to this stage so that I can share my views and my research to you. And I believe that the presentation that I will be sharing with you won't bore you enough. And I can give you some insights into the research that can be done by all of you. And I can share my opinions, views, and I can get some opinions and reviews from your end as well. So without wasting any time, I would like to begin. If my voice doesn't break and if I'm sounding pretty okay, then please, ma'am, do I have the permission to begin the presentation?
Yes, sir, sure. We are fortunate to have you today with us.
Yeah, thank you. So I will be just sharing my computer screen. I hope my screen is visible. If I'm good to go, then
yes, sir. Yes, sir.
Okay. So yeah, the topic for the day that I will be giving a presentation on is the proteases of SARS-CoV-2 as an attractive target for drug development. So if you all know that the SARS-CoV-2, the great pandemic, had shaken the world, and as a researchers like us, we are trying to find a solution to it. Not the major solution, but we are at least trying to put a little bit of effort into, we can help the society, or if we can help the researchers to gain some ideas regarding this. Okay, so now if we just see the SARS-CoV-2 and the ideal ways by which, just a second, the ideal ways by which we can, target and and combat this virus, there are two major strategies. The first strategies is very obvious, and I am pretty much sure that all of you have taken this vaccine, and without vaccine right now we wouldn't have been at this point. But so if I just break down, say 70% of the world which have resources were focused on developing the vaccine, and right now we have so many vaccines, and because of these vaccines we are pretty much safe. So we are very much thankful to this community of researchers who are working on vaccines that has bring us the light of hope that we can combat this this pandemic. Now the other groups of researchers which are basically working to combat this virus are trying to develop a drugs which can basically fight with this. Okay, so we, and in my lab and my students, we started working on this alternate approach. Why alternate approach? Because we don't have, as an institution, basically if we see, it is very difficult to directly start working on vaccines. But there are ways and there are methods by which at the university level, with the minimal amount of efforts, minimal amount of resources, we can basically study a lot of the biochemistry about this virus, and we can work on this alternate approach. Now, alternate approach means to develop drugs, to develop medications, or to propose medications which can work on this virus. Okay, so my basic research was this alternate approach, that how we can develop drugs which can combat the SARS CoV2 virus. Yeah, ma'am, am I audible, or my voice is not breaking, right? So any sort of confirmation? Okay, so I believe that my yeah,
audible.
Yeah, okay. So if we just focus on the structure of virus and the entire biochemistry of virus, there are two major classes of proteins which are found in this virus. Okay, the first family of proteins which are found on this virus are called as structural proteins, and one of the very famous one is the spike protein. So one in the green that you can see are the spike proteins. But apart from spike proteins, there are another proteins like small envelope membrane protein, that is a E protein, M protein, and then we have N protein. So all these proteins are there, which basically all the structural proteins give rise to the structure to the coronavirus. So these all proteins are basically responsible for giving the structure to this coronavirus. Okay. On the other hand, we have non-structural proteins as well, means the proteins which are very important for the life cycle of the virus. Without this protein, if we basically block any of this protein, what will happen? The life cycle of this virus will stop, and that is a good thing. So basically that is what we wanted to do. Now, out of all these proteins, the two major players or two major attractive proteins against which we can develop drugs to block their activity are these NSP3 proteins and NSP5 protein. So if we just see, NSP3 protein is also called as a main protease. So the name suggests it is a very important protease, and NSP3 is another protease which is also known as a peptide-like protease. So the major idea is that suppose if we can block these two proteins by taking any kind of a medication, then this virus will stop multiplying in our cells. Okay, so the idea is to develop inhibitors of these two proteins. Now let us basically we need to understand what these proteins do in the biochemistry of this virus. So the another name of this NSP5 main protease is also called as a MPRO. Another name of the same protein is 3CL-Pro. So whether if I say NSP5 or MPRO or 3CL-Pro, it means one and the same. On the other hand, this NSP3 peptide-like protease is also called as a PL-Pro. So MPRO and PL-Pro are the two major players or two major attractive protein targets against which we can develop drugs for. So now if I just show you the structure, the MPRO looks something like this. Okay, it is something like this, and basically this is a monomer, but in reality it exists as a dimer. I will show you that later. Okay, so now yeah, so basically what happens and why it is called as a main protease. So first of all, let us understand the basic genetics of this virus. So this SARS-CoV-2 is basically a RNA virus, and it has the genome size which is equivalent to around 30 kilobases. So this, the size of the RNA which is there inside the capsid of this virus is around 30 kilobases, right? So now what happens is that when this 30 kilobases are translated, it gives rise to two polyproteins. Matlab kya? Means this, when this RNA is a long RNA, if you read it from the particular reading frame, it will give rise to one polyprotein. And now if the ribosomes try to read the same RNA with a different reading frame, or like probably like frame shift, okay, it will give rise to another protein, a polyprotein. Now why this is called as a polyprotein? Because it is just the juncture or a mixture of all the different proteins. Now what happens is once these two polyproteins are developed, or it has been created by the translation of this RNA genome by ribosomes. So ribosomes will create these two polyproteins, and as soon as these two polyproteins are created, what will happen? This is then broken down at different 11 places. So as soon as it is broken down at different 11 places, it will give rise to all these different proteins. Okay, and this breaking of this polyprotein takes place by this empro. So that is why it is called as a main protease. So without this main protease, the polyproteins won't be broken down. And if these polyproteins are not broken down, we will not have the other NSPs that are required for the functioning of the this virus. So that is how it will work. Now NSP3 is again very important. So once this polyprotein is broken down, NSP3 is also being formed. So we will talk about this NSP3 later in this presentation itself. Now, what happens is that this is the structure of SARS-CoV-2. Okay, so this is the image that I have taken. Now, if I compare the same structure with other coronaviruses, which are also called as human coronavirus, okay, if you see the structure is very identical. Okay, so the structure of human coronavirus and structure of SARS coronavirus is probably identical. There are definitely mutations, but we are concerned with only two major amino acids over here, which is called as histidine and cysteine. So if you just see near my cursor, if I just zoom a little, so if you just see this part, histidine and cysteine, they are forming a catalytic diode. So this is a very important two amino acid. Without these two amino acids, or if we block this amino acids, or if we cap this amino acid, the entire protein will stop working. So these two amino acids are very important. Similarly, if you see the structure of SARS-CoV-2, what is happening in cysteine? If I just show you over here, is my screen visible? So if you just see the cysteine 45, 145 and histidine 41 is basically making a catalytic diode. The same numbers, if you just see for human coronavirus, the histidine is number 41. The same 41 number is over here, but if you see the cysteine in human coronavirus, it is 144, whereas it is 145 over here. Doesn't matter. But ultimately, they are forming this histidine, cysteine cysteine catalytic diode. Okay, so that is what the understanding, the basic fundamental lies. Now, so when we started working, we came across this research article which says structure of empro from SARS-CoV-2 and discovery of its inhibitor. So if you just see the volume, it is one of the volumes published in the year 2020. So it is a pretty recent article, and it was published in Nature. So I read this article and I came to know about the inhibitors that can be there for this protein. So same protein we took. So until this time, the crystal structure of SARS-CoV-2 was not identified. So this is the first paper, and these people first isolated and purified the SARS-CoV-2 main probe and submitted its 3D structure into this PDB. Okay, so I came across this article and I studied what basically it is. So now this is the image from the same paper, and it says that this is basically a dimer. So the red part, if you just see in the middle, is one monomer, and the blue part is another monomer coming together forming a dimer. So if you just see the structure over here, it is N3. So this N3 is a molecule which is basically sitting at the active site of the protein, and it is basically blocking the activity of this SARS-CoV-2, okay, or Mpro. Now this drug, which is N3, is right now in the clinical trials. And people have proposed, the researchers have proposed that this can strongly inhibit the activity of AMPRO, and hence we can use this as a drug for inhibiting the activity of main protease. So now if I just zoom a little, and if you just see what is basically happening, is that this molecule which is known as cysteine, this is the entire molecule in the green one, okay, it is known as a N3. If you just see over here, this entire green molecule is N3, and this N3 is basically interacting at this box which is saying cysteine 145. Okay, so cysteine 145, if your molecule is making an interaction with cysteine 145 in the active site of the protein, then we can say that this molecule have a tendency to inhibit this protein. Okay, so this is what the idea is. So I came across this, and then I came across another paper which is from PLOS Biology, and here they have proposed this many different inhibitors which have ability to interact with cysteine 145 of this AMPRO. Now I came across this, and I started working with some theoretical bioinformatics, and I did some simulation studies, and I replicated the same study which was done in the paper of Nature, not the in vitro part but the in silico part. Okay, so and this is what was there. So the N3 one, which was in the red, is the co-crystallized structure of N3, and the blue one is something that I have replicated in my computer lab. And I said, okay, fine, my algorithms are working pretty well, and I can identify certain inhibitors like which can basically bind to this protein in its active site and also interact with cysteine 145. Now, later what I did was that I created a small library of all the secondary metabolites from fungi, and I identified a molecule which is known as pyronigrin A. So this pyronigrin A had an ability to interact at the same active site to that of an N3, and if I superimpose both the inhibitors, so one is pyronigrin A and another one is the one which was published in Nature, I tried to superimpose them, and I found that it was making all sort of identical interactions with this pocket of the protein. And if you just see, this is the molecule in the blue is one of the phytochemical which is pyronigrin A. It is coming from the Aspergillus niger, to be very precise. And if you just see, it is making a very strong hydrogen bond interaction with the cysteine 145. And if you just see the structure is very small, and still it is making so many interactions in the active site. So if you take the ratio of surface area of this molecule versus the interaction formed, it is much greater than the proposed drug which is under the clinical trials. So now we can say that, okay, fine, fungus. Why fungus? Because if we see the past, we have isolated so many drugs from fungus. The first antibiotic basically came from fungus. Okay, most of the drugs for the cholesterol, it was identified from fungus. So can fungus also have some metabolites which can inhibit the activity of some viruses? And I propose that this molecule can be one of the molecule which can interact with the active site of this protein. But we don't have in vitro facility because it requires a very national laboratories can hardly work on this. So I propose it, but we can validate the results obtained by making some sort of a simulation. Okay, so another molecule that I identified was a flaveolin. So flaveolin and pyronigrin are the two molecules that are identified from the library of fungal metabolites that could bind to this active site. Again, if you just see the structure of pyronigrin, it was again making the interaction with the cysteine molecule, but this interaction was not through the hydrogen bond, but it was through the hydrophobic interaction. But anyways, so we propose these two molecules. And if you just see the structure, these two molecules are highly identical. If you just see, both of them have two rings, the size of the molecule is identical, and both could basically interact with cysteine 145. Now, we did some sort of simulation. Now, how many of you will be comfortable with the word simulation? That is, I don't know. But simulation is a methodology where you create an artificial environment. Like you create an artificial box. You fill your protein, your ligand, and your drug inside, and in that box you put some water molecules and you keep them or you relax them. As soon as you relax the protein and the drug, if the drug is supposed to bind to the active site of the protein, it will go and it will bind by its own. Okay, so that is the spontaneity of interaction can be studied between the drug and the protein. So these are few values, and for this we made use of the program which is known as a GROMACS, and this is all the reference molecule. It was making, say, how many interactions it was basically making. So if you just see, it was making the reference drug which was published in Nature, we took as a reference. And in my studies, it was making, say, around 6 to 7 hydrogen bonds or 6 to 7 total interactions, whereas pyronegrine A was able to interact up to 6 in certain cases throughout the simulated period. And the another drug was proposed metabolite was able to interact up to 8 interactions with this protein. And we found that, okay, this is very nice. And we published both these metabolites into 2 different research papers. So the first research article that you see on the top of biochemical chemistry talks about the molecule pyronegrine A, and the another research article talks about the flavulin as the inhibitor of MPRO. We continued our research, and I thought that we should also work on this PL Pro. So now, before proceeding further, I am giving the name Saga 2. Why? Because if you see the original slide, we have talked about 2 different proteins. One was the MPRO and another one was the PL Pro. Now, again, this is a very important protein. Why this is very important protein is because I have to show you some biochemistry of this molecule. So now, if you just see some animations, what happens is that as soon as the virus comes and it binds to the ACE2 receptor, okay, so this, what you see over here, the juncture, the spike protein is attached to the ACE2. As soon as the interaction takes place, the entire viral particle is taken inside and the viral RNA is released inside the cell. So the cell that you see, this one in the green, or say, I don't know which color to say, but it is a pista green color, whatever. So this green cell is the human cell. It is a representation of a human cell, and the virus is infecting it. Now, as soon as the viral goes inside, it will be converted into polyproteins as mentioned earlier, and those polyproteins will be broken down by main protease, and ultimately you will have this NSP3. Now, NSP3 is also called as PL Pro that I have mentioned over here. And now what does this PL Pro do is that it will break others polyproteins as well, and it will give rise to other structural proteins. The viral RNA will replicate inside, it will fit into this viral coat, and then it will be released. So this is one function of this PL Pro. So if somehow if you block this PL Pro, then basically the viral particle will not be able to it will not be able to capture the viral RNA inside the coat, and it will not be able to assemble itself properly, and it will not be able to go outside. So this is why this protein is of very importance. If you block this, the assembly of the replicating virus inside the host cell will stop. Now what happens is, now on the left hand side, on the right hand side at the bottom of the screen, I have shown the nucleus, and inside the nucleus I have shown the DNA. Now, ideally what happens that when any virus enters into the human cell, our body will recognize this virus as a threat and it will start producing interferons. Okay, but this PL Pro will stop this body, it will stop our body being identified by the virus, and it will not allow our body to release interferons. So it is the basic requirement, or innate immunity will not be activated if this PL Pro is there. So how does it do it? So this viral RNA is first being detected by some 2 proteins which is known as ISG15 and IRF3. So don't go into the details, but these are the 2 proteins which are there in our human cell. So this viral RNA will be detected by these 2 proteins. Okay, and as soon as this is detected, what will happen? This IRF3 will split from ISG15. It will form a dimer. It will bind to the DNA. It will go inside the nucleus. It will bind to the DNA, and it will activate the transcription of genes. As soon as this genes is transcribed, what will happen? It will produce more IRF3. And if this process takes place, it is going to basically activate interferon 1 gene. Okay. So now what happens is this PL Pro will basically come over here and it will degrade this IRF3. So this IRF3 is degraded by this viral PL Pro. As soon as it is degraded by this PL Pro, it cannot do this function and it cannot activate this interferon 1. And if interferon 1 gene is not activated, our body will not be able to produce interferon. So this PL Pro is doing two major functions. First, it is allowing the viral assembly, and second is that it is basically blocking the activation of innate immunity by blocking the interferon. So if somehow, if we can control or if we block the activity of PL Pro, then what will happen? We can protect ourselves from the virus. So हमें करना क्या है? We have to find inhibitor of this PL Pro somehow. And if we find some inhibitors of this PL Pro, then basically it is a good thing for us. And we can basically use that inhibitor as a drug. So as soon as viral particle goes into your system, you take the drug, it will block the PL Pro. Now the fun part about this both PL Pro and M Pro is that we do not have any analogous protein in our body which is identical to these two proteins. So there is very less chance of cross reactivity of drugs. Okay, so that is why they are chosen as the two most important viral protein targets. Okay, now after a lot of research, so the research on coronavirus basically started in the year 2008 with the first outbreak of SARS-CoV. So the first SARS was in the year 2002 or 2000, then MERS came. Okay, so in the year 2000, early 2000s, the research started, and we have already one molecule which is known as a GRL0617, and this is the Pro1 inhibitor of this PL Pro. So we can use this molecule as a reference molecule, and then later what happened? The SARS went away. People stopped working on this, and this drug remained in the clinical trials for a very long time, and people forgot about it. Okay, so basically this GRL0617 is a reference drug which is there in the clinical trials. Now, if you just see again one more article from Nature, and it says that this reference GRL0617, which was originally created for SARS, can also interact with the this PL Pro of SARS-CoV-2. So people opened the old books, they took this molecule and they tried to interact with this molecule, which is PL Pro of SARS-CoV-2, and they realized the drug which was originally invented for SARS-CoV can also work for SARS-CoV-2. So this is the paper which says that if you take GRL0167, then what will happen? It will bring back your innate immunity, and it will also regulate the replication of SARS-CoV-2, and it will block it. Now, regarding this GRL0617, now if you see the structure, it has a naphthalene moiety. So if you just see, there are two benzene rings attached together. So this part that you see is called as a naphthalene moiety. And the research says that molecules which have naphthalene structure inside the entire chemistry, then the molecule having this naphthalene moiety has capability to interact with the active site of this protein and inhibit it. So if you just see over here, GRL0167 has the ability to interact with tyrosine 268. Okay, so tyrosine 268 again for both the SARS-CoV-2 and this is the basic normal SARS. Okay, so now this was the original invention of this drug. If you just see it, it dates back in the year 2008, and this was originally invented for SARS. And people realized that this molecule which was originally invented for SARS can also work for SARS-CoV-2. Okay, so again what I did was I replicated the entire study once again. What I did was, this is the image, this is the image from my research article. So what I did was, I took the PL Pro from SARS and SARS-CoV-2, okay, and superimposed it, and also I superimposed the inhibitor, and I found that both the inhibitors were interacting in identical fashion in the same orientation. If you just see, in the blue one is the one protein and the red one is the another protein, and both these proteins have this ligand, and both the ligands are also superimposing on each other. and both of them are having this naphthalene moiety on the upper side and the single ring on the lower side. Now, this is the basic interaction, and if you just see this interaction, tyrosine 269, the interaction made by these molecules at this tyrosine 268 for SARS-CoV-2 and tyrosine 269 for the old SARS. Okay, so the number is differing by one amino acid, but that doesn't matter. We have to focus on this tyrosine. So as soon as the molecules, if you just see the naphthalene moiety, so this naphthalene group is basically interacting with this tyrosine residue in the active site by making a very strong hydrophobic interaction. Okay, so this was the original drug that was taken as a reference for my study, and I also took it. And then I did some study, and I again took the library of antifungal compounds, and I come across this molecule which is known as a fonsacin. And if you just see the structure of fonsacin, it has this 2-naphthalene group, and it is a naphthalene derivative. If you just go and if you see some, if you just Google it, and if you find this is the fonsacin molecule, which is isolated from the aspergillus, and to our surprise, it is making a very strong interaction with tyrosine 269. Okay, by making 3 different interactions, 1 hydrogen bond and 2 hydrophobic interactions over here. And again over here, again it is making identical interaction like the one which was shown by the reference drug GRL0617. Okay, so mind well, the molecules on the bottom are the molecules taken from the fungus. The molecules on the top are taken from the reference paper of Nature and Science. Okay, and I compared both these molecules with some in silico studies, and I found that our molecule, which was there in the library of fungal metabolites, it also has the naphthalene moiety. The size of the molecule was almost identical, and it also had the same affinity to interact with tyrosine 269. Further, to confirm this study, we did something which is known as a simulation. So again, I mentioned earlier, and if you just see the structure, okay, this is for the SARS-CoV-2, the bottom one is for SARS-CoV-2, and the bottom one is for fonsacin, whereas on the top one, the top box is for GRL0617. So this is GRL0617, and this on the right is entirely fonsacin. So now if you just see the arrows on the left hand side, proline 248, tyrosine 264, and tyrosine 268, these 3 amino acids is continuously making interactions throughout the simulated time. Throughout, if you just see the entire checkbox, it is all brown color. Now this brown color indicates strong interaction of these 3 amino acids with this molecule, and these are the amino acids on the y-axis. If you just see, okay, these are all the amino acids which are located in the active site of protein. Again, if you just see proline 248, constant interaction, tyrosine 264, constant interaction. One additional interaction of glycine 266 is also possible over here. If you just see, very strong interaction, and tyrosine 268, constant interaction again. Okay, so the pattern of interaction showed by both these drugs was also identical throughout the simulation, and the entire simulation in this case was performed for 100 nanoseconds, which is considered to be very large if you just see computational power. So this took around 1 week for the simulation to take place in the computer. And after simulation, I found that these interactions which was proposed by, say, docking studies were validated by simulation studies. And further, this was then published in this article in the Molecular Diversity. Then we continued our work, and then we said that, okay, fine, we have worked with the fungal metabolites, and we came across a database which is known as the NPAS database. And this NPAS database comprises of, say, 35,000 natural compounds from eukaryotes and prokaryotes. So it is a database containing all the phytochemicals, all the secondary metabolites, all the drugs which are coming from the natural sources. Okay, we did this high throughput virtual screening. We shortlisted 30 compounds which had ability to interact with tyrosine 268. Then we screened down to 6 compounds. Then we did some free energy change calculations. We identified 1 compound. We compared the drug-like properties, and we come across this molecule. I will just show you later. So this is just a graphical abstract that I've shown you. We came across this molecule which is known as, I don't know how to pronounce it. It is Cisla piniaphenol, cislapiniaphenol A. So if you just see the structure, this is a natural metabolite. And if you just see the structure, again, it has two naphthalene-like moieties, but here there is an oxygen, doesn't matter. And if you just see two rings over here in GRL 061, two rings, identical two rings over here, and at a terminal end, there is another ring which is over here. So if you just see, the structure are so much analogous. And it was able to interact strongly with tyrosine 268 over here. Very strong interaction. So this was one of the findings that we obtained. And again, we did the simulation studies. We compared it with this. So if you just see again over here, proline 248, 264, 268, and 269, same interactions, same intensity, or even better intensity was obtained for this molecule, which was there from the natural source. So we continued our research, and then we came across, we published this in this article. Then further, what we did was, we basically isolated all the different bacterial metabolites from the zinc database. From the zinc database, we identified we downloaded all the secondary metabolites known to be produced by bacteria, and we did the entire same thing again. And we then came across this molecule. I don't have the name for this molecule, but it has a zinc ID, which is 444-59905. Again, this molecule was able to interact very strongly with this tyrosine 268, if you just see. And this was a top compound from the zinc database, which was coming from the bacterial origin. Okay. Again, we did some simulation studies and compared with the reference drug, and it was obtained that proline 248, tyrosine 269, all these interactions, which were key interactions, were there. And then we published this in this article. Okay. So I would like to say that severe acute respiratory coronavirus is an agent emerging and re-emerging infection. So this article was basically published in the year 2007. And I like one very nice statement from this, that the authors have mentioned during the year 2007 that the SARS-CoV2 or SARS-like infections or SARS-like viruses are just like a ticking time bomb. Sooner or later, it is going to come and it is going to infect humans, and this was predicted way back in the 2007. And at that time, the authors from China itself, they had said that the possibility of re-emergence of SARS and other novel coronaviruses from animals or laboratories are therefore the need for preparedness should not be ignored. Means we should not take these viruses lightly. Even though the pandemics come and go, we should not stop working on this virus, because they will re-emerge. This virus has a tendency to come again and again and again, keep mutating, come again, and they have this tendency to create more and more and more deadliest viruses. So we should not stop working on this, and we should be very prepared whenever such new strain of coronavirus comes. So I would like to end my talk with this statement, and I would like to say thank you. And I try to, keep my presentation within 45 minutes, the stipulated time. So yes, ma'am, the session may be probably open for the questions.
Yes. Thank you so much, sir. You are always an application of inspiration, and our students are highly fortunate to listen such a advanced knowledge from you. And it all occur always whenever you come into the Parul University. So we are having some list of the questions from the different students.
Yeah, sure.
Yes, sir. Question number 1. As you explained, polyprotein 1A, 1B will be broken down by these pepin-like proteases, and then they will be giving rise to other NSPs also. So then how NSP3 and 5 will be produced by virus for the first damage just after the entry of the virus into the host cell?
Okay, very nice question. Now if you just see, when the viral particle comes inside, so I got the question as I am there. So let me just, okay, so I'm coming to this. So the question, as far as I believe, am I sharing my screen?
No, sir, it's not visible.
Okay, no problem. Yeah, so I will share my screen. So the
Yes, sir,
question,
it's visible.
yeah, so the question is that when the virus is infecting the human body for the first time, right?
Yes, sir.
We
Yes, sir.
don't have this
How
protein.
NSP3, sir, question is how NSP3 and 5 will be produced by the virus for the first damage just after the entry of virus into the cell?
Now, certain cases, what happens? Suppose, let me tell you, this polyprotein is not a very strong structure. Okay, so now it is like I will tell you, this polyprotein will be there in our body, produced by this virus, and somehow, some way, this polyprotein will have this slight amount of autocatalytic activity, and it will break itself. But once it breaks and produces one molecule of NSP3 or NSP5, it will just increase the speed tremendously. So, for instance, when the virus is infecting the human body for the first time, suppose it takes 15 minutes for the first polyprotein to break down itself and release a molecule of NSP5, it will take probably 15 or 20 minutes for auto degradation of this polyprotein in our system. But once the first molecule of NSP5 or 3 is released, by that time what will happen, a lot of these polyproteins would have accumulated in our system, and rapidly this NSP3 or 5 will start degrading all these polyproteins and it will start producing this molecule. So that is how it basically works. So initial kick is very important. If that doesn't take place, if initial 15, 20, 30 minutes when our body identifies this polyprotein as a foreign molecule and start degrading it, ओके उसके पहले अगर NSP3 और 5 बनना शुरू नहीं हुआ, then the polyprotein may be degraded, fine, doesn't matter. Meanwhile, more RNA will produce more polyproteins, and somewhere down the line, one of this polyprotein will break down autocatalytically, and it will produce one or two molecules of NSP3 that will simply aggravate the process.
Thank you so much, sir. The next question is from another student. What are in silico studies related to this topic?
Okay, so in silico studies, if I just show you, so I will just come to the one of the flow charts that I have shown you, or one of the so far. Okay, so let me just come back to this slide. Okay, so now first of all, we did multiple kinds of study. We started with the molecular docking, but it is not the ideal or the normal way of molecular docking. We did some docking which is called as a constrained molecular docking. Constrained molecular docking means we give the program the signal that you screen the molecules, but you give the outcome of only those molecules which have ability to interact with say tyrosine 268. So out of 35,000 compounds, I will not be doing the normal docking. I will give the program this constrained docking parameters where I can screen only those compounds which have ability to interact with tyrosine 268. Then the next thing that we will do is molecular dynamics, where we can basically study how ligand and how protein under the simulated artificial environment, how they can behave. And based on that, we will do this MMGBSA free energy delta G calculations, where we can identify the affinity of the ligand to interact with the protein. And that will basically give us the hint that at what concentrations the inhibitor may be binding to the or what is the affinity or the how easily or how effectively, what sort of attraction or how spontaneous the interaction is there. So that has been done with MMGBSA. Then we did some kind of post molecular dynamics assessment, where we can get the ideas like RMSDs and RMSF, types of interaction profiles, all those things. That is known as post dynamic simulation studies or assessments. And then we have to see whether the drug or the molecule that we have chosen, okay, is it actually having drug-like properties or not. So for that, we have to do something which is known as ADMET assessment. So ADMET stands for absorption, metabolism, then toxicity. What is the toxicity of the drug? How efficiently it can travel the blood-brain barrier or not? Is it serving as a toxic to liver? Does it have ability to inhibit some kind of uh, uh, क्या बोलते हैं cytochrome P450s and etc. And then we also did one more thing which is known as e-pharmacophore mapping, where we can identify or compare the structure of two different molecules based on the fundamental chemistry of the structure. So suppose if one molecule is aromatic, another molecule is also aromatic. So are there some features which are identical or matching? How many hydrogen donors does a molecule have? How many donors or acceptors does a molecule have? Then what is the overall surface area? Then what is the p-value of the drug? So all those things, all those parameters we compared. So basically in very short, it was constraint docking, e-pharmaco feature mapping, ADMET analysis, free energy change calculations like MMGBSA, drug-like properties with ADMET. That is the things that we do for this kind of a study.
Thank you so much, sir. The next question is, tyrosine 268 is good interactive site for phytochemicals, but how drugs will differentiate between the host and viral proteins?
Sorry, I didn't get the question. I'm tyrosine 268. So this one, okay,
yes,
it is. Yeah, so it is interacting. So therefore, we choose them. Now, what is another part of the question?
How drugs will differentiate between the host and virus particles.
Okay, so that is what I said, that the protein that we are choosing, this M-Pro and PL-Pro, we do not have any identical protein in our system. So basically, this drug will not have any binding affinity for the human proteins, because the protein, the active site of this, say, GRL0617, okay, for instance, the active site of this PL-Pro, the similar active site containing protein in human body is probably absent. And so the drug will not show any cross reactivity. So that is, I believe I have given justice to the question.
Yes.
So we do not have identical protein where this molecule can go and bind. That is my answer. Right. But yes, definitely human body is very complex. There may be chance that it can interact with one of the or two or three or different kind of cytochrome P450s, because it is very huge family. Ultimately, this molecule will then in our body, it will go to liver. And as soon as it goes into liver for detoxification, it will interact with one of the cytochromes, but it will probably, when we screen them, it should not inhibit their activity. It should be easily degradable. So that is that we have screened in the ADMET drug-like properties.
Thank you, sir. One more question.
Yes.
How proteins are better target size as compared to S protein and ACE2 receptors?
Okay. Now my this thing is, this ACE2 and ACE2 and spike are the worst candidates to design a drug for. You can use spike protein definitely to create the vaccine, but you cannot simply block. So what will you do? Will you puff the powder into your nose? So if suppose if a phytochemical is there, okay, and the virus is also going from your nose, how will you block or how will you coat the surface of the spike protein? Secondly, spike protein has a flat surface. ACE2 also has a flat surface, but they are binding together with the amino acid interaction. So protein does not have a cavity. And for any drug to work, it should bind to the active site. And if you just see the structure of spike protein and ACE2, there are research that people are also trying to block the flat surface. But blocking the flat surface, like a flat surface of say spike protein or the flat surface of say ACE2 receptor, okay, I don't think they are the good choice of targets for designing the drugs for. Yes, definitely we can use the elements of spike protein to develop a vaccine, because spike protein will go into your body. It is only a spike protein, not the entire virus. So it will activate some antibodies against it, and your body will definitely get some immunity. But if you want to design a drug, then definitely you should target only those proteins which are, number one, those proteins which are not identical in your human body. Number two, that they should be soluble enough. They should have the active site, and they should be accessible by the drugs. Now, suppose spike protein, as soon as the viral particle interact with the ACE2, the spike protein is of no use. So what will you do? And SARS virus will go from your mouth, from your tongue. So what will you do? How will you block them? Will you keep eating the phytochemicals throughout the time, and you will keep sniffing the phytochemicals so that the phytochemicals stay in your mouth? So I don't think that is a better idea. So therefore, a lot of research have gone into this, and therefore they have identified only these two proteins as the probable targets.
Thank you so much. One more question.
Sure.
What is the relation between the binding energy and potentially of phytochemicals?
Sorry, binding energy and
Potency of the phytochemicals.
very nice question. Okay, so binding energy is just the energy which is calculated by the algorithm. Okay, so now for instance, binding energy does not always mean that the drug will bind to the protein. For that, you have to identify the free energy chain. So the entire, if you put a drug over here in the protein, how the free energy changes are taking place, you have to basically calculate that. And if the free energy chain is favoring the interaction, only then the interaction will take place. So even if a compound is giving a very high binding score or docking score, it is not a surety that it will go and it will bind to the protein efficiently in the human system. For that, you have to see how the spontaneity of the reaction is, because the docking algorithm will basically put the drug over there in such a way that there is minimum torsion, there is minimum there is minimum of instability. But when you put the entire protein ligand complex in the water system, and does the ligand stay inside the cavity or just moves out? Because in the system we also have water molecules, we have water outside. So those hydrogen bonds created by water to the ligand can pull the ligand outside. So for that, it has to have very strong free energy chain in the negative values. More the free energy change in the negative values, that is through MMGBSA calculation. So that is 50% validation. And then the rest 50% of validation, whether the docking predictions will work or not, for that you have to do simulation.
Thank you so much. One more question, sir. You might be thinking that you are getting a lot of questions in the lecture what you have provided.
No.
So we are actually very fortunate to have you, and we are not losing this opportunity to interrogate you.
Yeah, very much glad to. Yeah, sure.
Sir, what are the difference between main and peptide-like protease?
Oh, main protease and peptide-like protease, they are completely different. Main protease is the main protease, which will basically is the main responsible for cleaving most of the proteins. So you just see the structure. If I show you, am I sharing my screen? Yeah. So yes. Main thing is PL Pro, which is a peptide-like protease, is responsible for only cleaving this NSP1, NSP2, and NSP3. Now NSP1 itself is a main protease. Okay. Now NSP2 and NSP3, so it is cleaving entire polyprotein at only 1, 2, and 3 places. Only 3 places out of 11. So out of 11 cuts, only 3 cuts are being made by PL Pro. Rest all other cuts are made by main protease. But PL Pro also breaks down the transcription factors which are required for the activation of interferon gene. So main protease does not do that. Only PL Pro does the inactivation of the transcription factors that are required for the activation of interferon gene. So that is what the major difference is. So yeah, in very short terms, main protease will cleave the protein at 8 different sites of the polyprotein, whereas PL Pro only cuts at 3 different sites. And NSP3, or that is a PL Pro, will cleave or degrade the transcription factors required for the activation of interferon, which is not done by a main protease.
Okay. Thank you so much, sir. One last question. Natural remedies we have used during the COVID infection, do they have any role in the inhibition of these proteases?
we cannot say because we do not know. So for example, I will give you the best example. Suppose if there is a plant, it is producing a phytochemical. Okay. Now it is reported that the particular plant is making a particular phytochemical. But what if that particular phytochemical is not been synthesized by the plant in say in the month of say summer, and you are taking the plant from summer. Okay. So for example, आपने सबने सुना होगा कि रात रानी, it will only produce that molecule at night. Okay, so you will identify only that molecule at night. So when we are taking natural products directly, we don't know what we are exactly consuming, which kind of phytochemicals we are consuming. Are we consuming the same phytochemical, same dosages every time? Okay. The second thing is we need to understand the profile of those molecules in their ability to interact with these molecules. So here when I'm saying I screened say 30,000 or 40,000 phytochemicals, and I got only 1 or 2 phytochemicals which are good. Now, when you are eating plant or the dried powder of the plant, it is rich in only 1 or 2 phytochemicals at a very large concentrations. Whereas other phytochemicals which are probably very efficient are in very minute amounts. So if you eat more plant to get more phytochemicals, probably those phytochemicals which are very large in concentration will damage your liver. For example, giloy. So if you just see giloy, if you keep eating more giloy for 6 months, it will definitely going to damage your liver. People are keep eating, they are just they are decoctions of this phytochemical powders and plants, and that is not basically a good idea because you don't know the composition of that. So in order to take one important phytochemical which is just present in minute amount, and when you're taking plant, you are taking other phytochemicals in very large amounts, and those large amount phytochemicals can damage your liver, definitely true. So without understanding the profile of the phytochemical, we cannot say that it is the and people always assume and keep a very fine line between say allopathy and Ayurveda. But if you see the allopathy is developed from Ayurveda. I'll give you an example. Aspirin. Aspirin basically is found in the plant. You simply extracted it, made a tablet, and now it is a part of allopathy. So we should not bifurcate this allopathy and Ayurveda together, because Ayurveda people do not have enough knowledge. In allopathy, at least if it is not perfect, good enough. It is not perfect, but at least we have the idea, much better idea regarding how the drug will interact with other drugs, how it will interact, what are the side effects. We have clear detail about it. So we should not basically put this line that, okay, Ayurveda is on this side and allopathy is on this side. Most of the drugs are coming from this side to this side. And we are taking a tablet and selling it into the market. Okay, aspirin, वो कौन से पेड़ से आते हैं वो पता नहीं. Even if you see tobacco, लोग क्यों खाते हैं? क्योंकि उसमें compound होता है. So as soon as you extract the tobacco, make a medicine, it will become allopathy. And that tobacco plant, if you are consuming directly, that is becoming Ayurveda. So there is a very thin line, and people should understand that when we are talking about phytochemicals or molecules or whatever, there is a very thin line between allopathy and Ayurveda. Though Ayurveda is much more sound, but I don't know how many people have very strong knowledge about it. But allopathy, whatever is there, even if it is not perfect, appreciated, not perfect, but at least what information we have some information. That if a person is having a liver damage, this drug should not be taken. But for Ayurveda, we do not have that kind of information. We might be giving some Ayurvedic medicines to some people who are having kidney damage, liver damage, and we are damaging it even further. So yeah, it's on both the sides.
Right, sir. Thank you, sir. Thank you so much. After listening your answers, students have created a lot of curiosity in the same topic.
Oh,
so thank you so much for your wonderful answers.
Thank you so much. Thank you so much.
Sir, you are an epitome to millions of Indians, and
oh my God.
inspiration
No.
of your
no.
your entire life is an inspiration, actually,
Madam,
being in such a young age.
Nothing like that. Yeah.
So being in such a age, actually, students should learn that what the Indian youngsters actually they can do. So they are highly, impressed by you, and the talk is very wonderful. I would like to pay my sincere greatest
Thank you so much,
gratitude to you.
But there was a lot
and
of
on behalf
exaggeration.
of entire
I would like to be addressed more humbly.
Institute of Applied Science, I would like to give you thank you.
Thank you so much, man. Thank you so much.
Thank you so much, sir. We expect that in future our students can get knowledge from you physically, not by the
Oh,
virtual.
definitely, definitely, sure, sure, sure.
Thank you so much, sir.
Most welcome. Yeah, thank you. Thank you for calling me to this webinar series. Thank you.